Good afternoon, everyone. My name is Mikhail Marcio, student ID 250100068 from the Biomedicine Program, and today I'll be presenting my scientific review paper entitled The Antibacterial Efficacy of Manuka Honey Against Biofilm Forming Chronic Skin Wounds. This review focuses on evaluating the potential of Manuka honey as an antibacterial and antibiofilm agent in chronic skin wound. The topic is particularly very relevant because chronic skin wounds infection remain a major clinical challenge in hospitals, especially with the increasing prevalence of antibiotic resistance. Back into this review, I aim to discuss the mechanisms behind Manuka honey's antimicrobial activities, summarize its findings from recent studies, and evaluate its potential role in modern wound care. Chronic wounds are wounds that fail to progress through the normal stage of healing within an expected period of time. Unlike acute wounds, which generally heal within weeks, chronic skin wound may persist for months or even years. Common examples include diabetic foot ulcers, venous look ulcers, and pressure ulcers. These wounds represent a significant healthcare burden because they are often associated with persistent infections, prolonged inflammation, reduced quality of life, and increased treatment costs. One of the major factors contributing to the delayed healing is the formation of a bacterial biofilm. Biofilms are highly organized communities of bacteria enclosed within a protective extracellular matrix. This matrix acts as a barrier that shields bacteria from antimicrobial agents and boosts the immune system. As a result, bacteria living within biofilms are much more difficult to eliminate than free-floating bacteria. Biofilms associated infections can persist despite treatment and often contribute to chronic inflammations and delayed tissue repair. Another challenge in wound management is the growing problem of antibiotic resistance. Pathogens such as methicillin-resistant Staphylococcus aureus or MRSA have become increasingly difficult to treat using conventional antibiotics. Because of these limitations, there is a growing interest in alternative antimicrobial therapies that can effectively target biofilms within reducing the risk of resistance development. One of the promising candidates is the Manuka honey. What is Manuka honey? Manuka honey is a type of medical grade honey derived from the nectar of Leptospermum scoparium, a plant native to New Zealand. Although honey has been used in traditional medicines for centuries, Manuka honey has gained particular scientific attention because of its unique antimicrobial properties. Unlike regular honey, contains high level of methyglioxal, commonly known as MgO, which is considered one of its primary bioactive compounds. In addition to MgO, Manuka honey possesses several characteristics that contributes to its antibacterial activity. This includes a naturally low pH, high pH osmolarity, reduced sugar content, and the ability to produce hydrogen peroxide. These properties work together to create an environment that is unfavorable for bacterial survival. Because of this combination of antimicrobial mechanisms, Nicani has demonstrated activity against broad range of wound-associated pathogens, including antibiotic-resistant strains. As a result, it has been incorporated into modern wound dressings and is increasingly being studied as a therapeutic option in wound care. For the mechanism of action, the antibacterial activity of Manuka honey is multifactorial, meaning that it works through several mechanisms simultaneously rather than targeting a single bacteria process. This is one of the reasons that it has attracted attention as a potential alternative to treatment. The first mechanism involves its high sugar concentration, which creates osmotic stresses. Through osmosis, water is drawn out of bacterial cells, causing dehydration and reducing their ability to survive and reproduce in the body. The second mechanism is its naturally low pH, which inhibits bacterial growth and interferes with the essential metabolic process inside bacteria. Another important mechanism is the production of hydrogen peroxide. Hydrogen peroxide generates oxidative stress, which damages bacterial DNA, proteins, and cell membranes, which contributes to bacterial cell death and limits the bacterial proliferation within the wound environment. That's one of the most unique features within Manuka honey is the presence of methylglyoxal. Methylglyoxal can interfere with bacterial protein functions and disrupt key metabolic pathways, further reducing bacterial viability. These combined effects weaken bacteria through multiple routes at the same time, and in addition to direct antibacterial activity, several studies suggest that Manuka honey may interfere with quorum sensing, which is a communication system used by bacteria to coordinate behaviors such as virulence and biofilm formation. By disrupting these signaling pathways, Manukani may prevent biofilms from developing properly and reduce their structural stability. This is particularly important because biofilms are one of the primary obstacles to successfully treating a chronic wound infection. To evaluate the current evidence regarding Manuka honey and chronic infections, a structured literature review was conducted. Relevant studies were identified through several scientific databases, including PubMed, Google Scholar, and Science Direct. These databases were selected because they contain large collections of biomedical and clinical research publications. And the literature search we focus on studies published between 2015 and 2025, and to ensure the review that included was recent and relevant findings. Keywords used during the search process included terms such as Manuka honey, medical grade honey, biofilm, chronic wounds, antibacterial activity, MRSA, Pseudomonas aeruginosa, and wound healing. Boolean operators such as and or were used to refine search results and improve the relevance of finding identified studies. Studies were included because if they were investigate the bacterial or antibiotic properties of Manuka honey and involve clinically relevant wound pathogens. Both laboratory-based investigations and clinical studies were considered after screening titles, abstracts, and full text according to predefined inclusions. This and the exclusion of and a couple of criteria. These studies were selected as for the final review data from these studies. Were then grouped into themes, including antibiotic activity, antimicrobial mechanisms, and clinical outcomes. The findings of this reviewed studies consistently demonstrated significant antibacterial and antibiofilm activity of Manuka honey against several major wound-associated pathogens. The most commonly studied organisms are Pseudomonas aeruginosa, Staphylococcus aureus, including MRSA strains, and Proteus mirabilis. Studies involving Pseudomonas aeruginosa reported substantial reduction in biofilm, biomass, and bacterial viability following treatment with Manuka honey. This finding is important because Pseudomonas aeruginosa is known for its ability to form highly resilient biofilms and is frequently associated with chronic food infections. Similarly, studies involving Staphylococcus aureus and MRSA reported reduced biofilm formation and suppression of gene expression related to biofilm development. This suggests that Manuka honey may not only kill bacteria directly but also interfere with the biological processes required for biofilm formation. Research involving Proteus mirabilis demonstrated reduced bacterial viability and decreased biofilm formation. Collectively, these findings indicate that the antimicrobial activity of Manuka honey is broad spectrum rather than limited to a single bacterial species. An important observation from the review literature is that positive effects were consistently reported across multiple studies and different bacterial species. Furthermore, none of the included studies reported complete resistance to Manuka honey treatment. This is particularly encouraging in the context of increasing antibiotic resistance and highlights the potential value of Manuka honey as an antimicrobial agent. The image shown in the slide illustrates a wound healing progression following treatment with Manuka honey in a veterinary clinical settings. Although the examples involves an animal patient, specifically a dog, rather than a human patient, it still provides a clear visual demonstration of tissue recovery and wound improvement associated with Manuka honey therapy. The wound gradually decreases in size, inflammation becomes less apparent, and the healthy tissue regeneration becomes visible over time. These clinical observations support the idea that by reducing bacterial burden and disrupting biofilm, Manuka honey may create a more favorable environment for tissue repairs. As a result, wound healing can proceed more efficiently, which provides in reducing recovery time and improving patient outcomes. Despite the promising findings presented in this review, several limitations were acknowledged. First, a significant proportion of the available evidence come from vital laboratory studies. While these studies are valuable for understanding the antimicrobial mechanisms of the honey, they cannot fully replicate the complexity of real wound environments. Factors such as host immune responses, tissue oxygenation, wound exudate, and the interaction between multiple bacterial species are difficult to reproduce under laboratory conditions. Another limitation is variability among clinical studies, differences in wound type, patient population, and treatment protocols. And outcome measurements can make direct comparisons challenging. In addition, many clinical studies involve relatively small sample sizes, which limits the generalizability of their findings. Unfortunately, variability in Manuka honey products also represents a challenge. Different products may contain different concentrations of methylglyoxal and other active compounds. As a result, treatment effectiveness may vary between products, making standardization an important consideration for future researches and clinical implementations. Therefore, although the available evidence is encouraging, the findings should be interpreted with caution, and further high quality clinical research is needed to strengthen the evidence basis. In conclusion, the evidence reviewed in this paper indicates that Manuka honey demonstrates significant antibacterial and antibiotic activity against major chronic wound pathogens, including Pseudomonas aeruginosa, Staphylococcus aureus, MRSA, and Proteus mirabilis. Through a combination of mechanisms including osmotic stress, low pH, hydrogen peroxide production, and methylglyoxal activity, Manuka honey is able to inhibit bacterial growth and disrupt biofilm formation. This reviewed study consistently reported reductions in bacterial viability and biofilm development, while clinical investigations demonstrate improvement in wound healing outcomes such as faster wound closure and reduced infection rates. And these findings suggest that Manuka honey has a strong potential as an alternative or adjunctive therapy in chronic wound management. However, despite these promising results, further large-scale clinical research are required to confirm its effectiveness and establish standardized treatment protocols. Overall, Manuka honey represents a very valuable natural therapeutic options that may contribute to addressing the ongoing challenges of chronic wound infections and the antibiotic resistance within bacteria. That is all for my presentation. Thank you for listening.